Hi folks, this is John Drasba from Biology 2, Mrs. DeWall-Malafite's class, and I am here to tell you about what my team and I investigated as our research project this spring 2020 semester. Our project was on tardigrades, but more on this later. Before we get into the project, let's go ahead and meet the team. First, there's James Fink. Next, there's Krishna Paul. And finally, there's me, John Drasba. Oops, wait, here's the correct me, not in costume.
Introduction. Finding a problem. Tardigrades are extremely resilient organisms capable of surviving otherwise unlivable conditions. However, they pose a severe weakness to high temperatures. Temperatures around 37 Celsius, for example, in a normal state will prolong in the eventual death of a tardigrade. This presented a problem that we wish to answer, and that is there a way to improve on this nearly indestructible organism through the use of acclimation to high temperature.
So we asked whether temperature acclimation could provide a thermotolerance increase to tardigrades, and this led us to our hypothesis.
Now we move to discuss the hypothesis of our experiment, in which we proposed that if tardigrades are exposed to temperatures outside of their normal temperature ranges gradually, and for long periods of time, they will become tolerant to such temperatures, and in turn their normal temperature ranges will increase. Based on the evidence uncovered in past studies, we did expect to see evidence of conditioning. After deciding on the appropriate method of conditioning the tardigrades, a trial-based conditioning system was developed to acclimate the tardigrades, as well as test the success, or lack thereof, of the conditioning in order to test the proposed hypothesis. Now we can see the process in which we actually underwent to supposedly condition our tardigrades. First, what we did is we took the culture in which we were provided with and took a quarter of the culture and set it aside to make sure that we had a control to test against. Then the other 3/4 of that culture was then heated in a water bath gradually to figure out at which temperature were the tardigrades showing signs of not being able to handle. Basically, we weren't looking for whether or not the tardigrades were dead or not. Based upon the asterisks down at the bottom, it showed signs, or they showed signs rather, of a state of shock or lack of activity. This basically proved to us that it were those temperatures that they were no longer able to handle. After determining the pinpoint temperature, we then took that pinpoint beaker and divided half of that into a condition 1 beaker. That condition 1 beaker was then heated for 45 minutes at a little bit of a lower temperature than the pinpoint temperature previously found, for about 45 minutes, and then we continuously microscopically observed to see whether or not they were slipping into a shock stage once again. Luckily, we found that they were not, so we continued with the conditioning process. After that, what we did is we went and we conditioned them again within a condition 2 culture. What that was just half of condition 1. So essentially, half of the condition 1 culture was being double heated, or double conditioned. For another 45 minutes, we repeated the same exact process. Once our conditioning was complete, we then tested every single culture against the unconditioned culture at the previously determined pinpoint temperature to see if we showed any evidence of an increase of temperature tolerance within the tardigrades.
As you can see on this slide, the left-hand picture shows the tardigrades in the water bath heater. Here we are taking tardigrade samples at various temperatures, placing them on slides and examining their activity levels, both under the microscope directly and with the microscope camera display. We performed the testing to see how they behaved and acclimated to various temperatures. Here are our results. There was an important finding in that one conditioned tardigrade remained active at the pinpoint temperature of 34 degrees Celsius. Additionally, many qualitative observations were made during the conditioning process, such as movement of the tardigrades and signs of movement. There were also new discoveries made from the observations.
Now to conclude our research. Not many tardigrades were able to maintain activity at the pinpoint temperature, even after conditioning, and those who did proved statistically insignificant. Even though we lack statistical significance, the hypothesis was supported. Our question might not have been answered directly, although possible new discoveries were made on the topic of tardigrades, as well as a new understanding of their adaptive abilities. This being said, more research is needed into the acclimation of tardigrades in order to answer the proposed question. Finally, consultation with professional tardigrade researchers is needed to confirm or refute the discoveries made.
I hope you enjoyed our presentation. Please let us know if you have any questions, and feel free to check out our poster and PowerPoint. And to Mrs. DeWall Malafite for all the knowledge and experience you gave us. Thank you.